So Wayne has been a scientific advisor for us from early in the program, and he's faculty leader of the Biomedicine and Biotechnology faculty group at Arizona State University. And past that, probably doesn't need any further introduction from me. So Wayne, I'd be delighted if you would introduce our speaker today, Judith, and then lead on to Judith. Wayne, over to you.
Okay, very good. So am I sharing yet? Oh wait, I have to.
You have to press some buttons to do that.
I have to press some buttons to do that.
Can I just remind everybody to go on to mute to make Wayne's presentation clearer? Thanks. Okay, go.
Great. It is my great pleasure to introduce Professor Judith Klinman today. And her talk is entitled "At the Interface of Quantum and Classical Behavior in Nature, Looking into the Eye of Quantum Tunneling and Enzymes and the Role of a Noisy Environment." She's in the departments. She started out in the Department of Chemistry, but now she's in the Department of Chemistry as well as Molecular and Cellular Biology. And she's also in the California Institute for Quantitative Biosciences at the University of California, Berkeley. So Judith was kind enough to send me this paper that she published in the Annual Review of Biochemistry. It's unlike most annual reviews that appear in that journal, where it's about not only her scientific contributions, but about how the stages of her life impacted those, moving through barriers in science and life. And so anyway, I found it a very, very interesting read. And I think that in this day when I see a lot of younger women coming into science, that I strongly want people to read it. So she grew up in the Philadelphia area and got a scholarship to the University of Pennsylvania, where she got her undergraduate degree in 1962, and had her first research experience at the Johnson Foundation at Penn, where the Johnson Foundation was run by Britton Chance, but Britt had brought together a large number of just extraordinarily talented people, including Ron Estabrook and Helen Davies, with whom she worked. And so to give you some sense of what an extraordinary experience it must have been for an undergrad to work there, at the time, Britton Chance really dominated the area of biochemistry at the interface of biophysics, and certainly with area of mitochondrial oxphos and photosynthesis. And at one point, people made the joke, a lot of things in science are discovered by luck, the rest are discovered by chance. And so, I can tell you lots of Brit stories that are very good, but anyway, so, just to say that it was, what a great way to start to be introduced to science. So she went on to New York University, where she got her PhD in 1966. And I put these dates down because, in fact, she didn't, it looks like it was 4 years, but it was actually 3 years. And you had your first child during this time, which makes it even more extraordinary. And so she did that with Edward Thornton, one of the pioneers of mechanistic enzymology, which was really in the development at that time, of which, of course, Britton Chance was at that interface as well. Then off to the Weizmann Institute for postdoctoral studies with David Samuel, and a subsequent apprentice with Charles Vernon, both of which related to a project testing Mitchell's chemiosmotic hypothesis for ATP synthesis. And Peter Mitchell was quite a character. And he had come up with his chemiosmotic hypothesis that membrane potential and pH gradients were coupling ATP synthesis to the electron transfer reactions, and people did not believe it. In fact, there were people who still didn't believe it after he got the Nobel Prize for it. So anyways, at that point, she returned back to Philadelphia to work at the Fox Chase Cancer Research Institute with Ernie Rose. And she stayed there for a number of years, where she essentially grew out of Rose's lab to become independent and essentially equivalent to an assistant professor. Then finally, her last move was to UC Berkeley, where she joined the chemistry department as an associate professor in 1975. And in your chapter, you wrote that you didn't really respond to a job position advertisement like most people would, but instead you just wrote letters saying, hey, I'd like to come work there, which is, I think, extraordinary. But having now also looked at your CV and seeing all the things you've accomplished, I'm not surprised they wanted you. But I include this photo of you with the Department of Chemistry members, and I recognize a few of them in there. I see Ken Sauer, and I think I see Melvin Calvin, Nobel laureate. And if you look carefully, you will all see that there is only one woman there. So it is Judith. So anyway, in your career now, as it has developed, you have more than 250 peer-reviewed publications, many more book chapters, news and views, abstract, and many, many awards. That goes on for pages. It's almost like, wait a minute, which one didn't you get? And so some of the notable ones are you're a fellow of the Royal Society of Chemistry, you're a member of the National Academy of Sciences. I saw you got the Mildred Cohen Award, but you didn't get the Ernie Rose Award. We have to do something about that. That's one that's missing. Anyway, but there's very few that are missing. But there's one in particular that I think that it's really very important, and you got the Presidential Medal of Science from Barack Obama. And I had to go on the internet to get this picture where he's actually putting the medal over your neck, and there you are with your family, which ended up you had 4 kids all total. So you managed to have a great career and children. People say you can't do it all, but it seems that you have. Anyway, I will reiterate, your seminar is At the Interface of Quantum and Classical Behavior in Nature, looking into the eye of quantum tunneling and enzymes and the role of a noisy environment. Judith, welcome.
All right, so let's see. Okay. Wayne, thank you for that. I'm a little overwhelmed by your introduction. I hesitated before sending you a copy of that chapter, because I wasn't sure how much I wanted to vote. You did a beautiful job and made me feel much more important than I normally do, but thank you for that. It was wonderful. And thank you, Jeffrey, and everyone for this opportunity. All right, so let's just get started. It's not advancing. This first slide is courtesy of my colleague Birgitta Whaley at UC Berkeley, and she's a theoretician very interested in the area of quantum biology. And what it is an overview of the various systems that have been interrogated, and you've heard many of them already within this symposia series. But I just want to point out the role of coherence, this question of how coherent is a process, and can we then ascribe the behavior to true quantum biological behavior. You've heard, of course, about the role of the light harvesting chromophores in photosynthesis, and the possibility of vibronic coupling between vibrational and electronic states. Let me just see if I can get the pointer. let's just see if I can get the pointer going. Yes, I hope you can see where I'm pointing well. There's also many fascinating issues in this first slide. There's the question of whether birds have the capability to sense the Earth's magnetic field through correlated spins. On and on it goes. Tunneling is mentioned in Birgitta's talk. This is electron tunneling in the context of how animals smell. And what I want to do today is expand our understanding of tunneling in biology to cover nuclei, in particular hydrogen and its isotopes. Now, proton-coupled electron transfers are highly prevalent in life processes and very fundamental. And on this first slide, I'm showing someone consuming their glucose stores to enable this young woman to run. And what's going on in this process is the oxidation of a reduced substrate to generate a reduced intermediate cofactor, which then must undergo a series of proton and electron transfers to generate the energy currency of the cell, which is ATP. So the prevalence of PCET and its fundamental nature is something that has caught our attention and led to many, many years of research. Now, this next slide is a discussion of the de Broglie wavelength. And the reason I included it is you may ask, well, we know the electron is small, it has a very long wavelength. Of course, it can move as a wave over long distances. But why should we think of the nuclei, hydrogen, deuterium, tritium, with the same behavior? Because if we just describe an energy of 4 kcal per mole to the system, you can see the wavelength of the hydrogen and its isotopes is really much smaller than that for electron. But the key idea is how far must the particle transfer under conditions of reactivity, and how does that relate to the wavelength of the particle? And in fact, when you transfer hydrogen between a donor and acceptor site, the distance that it transfers through computation primarily is in the range of 0.7 angstrom. So it's a correspondence between the distance transferred and the wavelength of the particle that makes hydrogen tunneling so compelling. Now, you all know about Schrodinger's famous book, which is What is Life?, where he questions whether quantum mechanics can truly be a part of biology. And of course, perhaps played a role in driving biological fitness, which is what I want to discuss at the very end of this talk. What I want to emphasize is that through our work and the work of many other laboratories, there really is extensive evidence now that there's nuclear tunneling in biological reactions. And this is just a cartoon, basically, of the fact that when you move through the barrier rather than over the top of the barrier, you have some very large potential catalytic advantages. Now, what I want to focus on for the majority of this talk is the class of reactions in which we're going to remove a hydrogen from a stable position so we can label it and understand its properties. And in this case, it's going to be carbon. And the acceptor can vary from another carbon to an oxygen, nitrogen, et cetera. And what I've just shown you are overlapping potential energy wells. And we have the 0-point energy vibrational effects within these electronic potential energy surfaces. And I'm showing that we can write wave distributions between the two wells, where protium has not only a higher vibrational level, but it also has a more diffuse wavelength because, in fact, its mass is half that of deuterium, which has a shorter wavelength and is going to encounter much more difficulty in transferring from a reactant to a product well. And this comes up repeatedly in the properties that we see for enzymes. Now, I also want to point out that most of enzymology has been discussed in the context of reducing a barrier height, and all of a sudden we're becoming very interested in the role of barrier width and the ability, of course, to get close enough for tunneling to occur, but also note that for coherence to occur in this tunneling reaction, we have to have transient energy levels between the donor and the acceptor wells. That will come up as well. Now, enzyme catalysis has been studied, and it's a rich, deep area, but the one true goal of being able to reproduce the enormous rate accelerations that enzymes bring about has been quite elusive. De novo protein design has been trying to do that, but without success in terms of the extreme end of rate accelerations. And I here summarize some work by Richard Wolfenden, where he looked at both small molecule reactions and the same reaction catalyzed by an enzyme. And he was able to estimate that the rate of acceleration of a given process by an enzyme can be as high as 10 to the 30-fold. This is more than the number of stars in the universe. And there are many efforts continuing, including directed evolution, which can accomplish some of this rate acceleration, but certainly not all of it. So we're trying to get at the physical properties that determine catalysis as a parallel line of investigation in my lab to go with the quantum biology. Now, proteins are quite large. An average size of a protein would be 40 to 100 kilodaltons. But what I want to show you here, the gray is the whole protein, is how small the active site of the enzyme is in relation to the full size of the protein. And for decades, the scaffold was considered basically a rigid framework that allowed these substrates, which are colored yellow and red, to tuck themselves into the active site of the protein. Well, today we're going to turn, in fact, to the scaffold and try to understand the critical role it plays in promoting the active site chemistry, in particular, hydrogen tunneling. So small molecules have been studied extensively that transfer hydrogen. It's one of the fundamental chemical reactions that occurs in both chemistry and in biology. And this is from a very important book published decades ago by Rodney Bell. And what he did was he looked at the temperature dependence of hydrogen transfer as a function of temperature. He compiled a lot of data and then introduced very useful concepts that could be applied to understand what was going on. And in this graph, you see that if we go over a wide temperature range, this would be the transfer of protium on the top, the transfer of deuterium on the bottom. And at very high temperature, where all the vibrational modes converge, so all the vibrational modes, the H and D vibrational modes converge to a single energy level, there's basically no difference in rate. And then most of the reactions that have been studied would fall into, above and below room temperature, which is of course where biology functions, and the lines were expected to give different slopes for protium and deuterium. And that's the origin of the isotope effect in this semi-classical model. But then as you go to increasingly low temperatures, people began to see some strange behavior, which is that the slopes of the lines have leveled out. The slopes, in fact, become the same for protium and deuterium. And the isotope effects become huge, maybe 1,000. And this was attributed to tunneling. And so this behavior of essentially no difference in energy of activation between protium and deuterium was first observed at these near absolute zero temperatures, very cryogenic, and has been considered a characteristic diagnostic of hydrogen tunneling. Well, when we looked at enzymes, so we started looking at these phenomena because of discrepancies in the interpretation based solely on 0-point energy differences. And we started to pick up a very weird phenomenon that we couldn't understand at the time. It took a while to develop the theory. And that is within the temperature range that biology functions, when we measured the rate as a function of temperature, we were able to get very reproducible data under conditions where chemistry is rate limiting, and the slopes were found to be parallel to one another, indicating, in fact, that under the conditions of room temperature, biological CH activation, with native enzymes, the energy of activation for D didn't look very different from the energy of activation for H, which is exactly what Bell had indicated for the very low temperature behavior of small molecules, with the exception that, of course, the enthalpy of activation is not zero. For enzymatic reactions, it can be very large. So we have this combined effect of no difference between the slope for deuterium and protium and large activation energies. And this has been one of the important observations that has led to a lot of theoretical development and further experimental testing. And this is just a summary of some of the systems where the isotope effect at 25 degrees and then the intercept, which reflects the difference in the slopes, is not unity as it would have been predicted to be, but is very close to the measured isotope effect. Now, in all these instances, there's a large deviation from an intercept of unity, indicating that, but the AH over AD values don't always equal the isotope effect, indicating that there is not only a regime where there's absolutely no difference in energy of activation for transfer of D versus H, versus a regime where there is some small difference. And so that then leads to some discrepancy between the single temperature isotope effect and its temperature dependence. But this is a widespread phenomenon. So this then raises some major questions. First of all, how can enzymes facilitate quantum tunneling with behaviors that are thought to, with the kind of behavior that was thought to be restricted to very low temperatures? Okay. Second, enzyme reactions are generally quite temperature dependent. Average energy of activation would be 10 kilocalories per mole, but wave function overlap is not. So how do we uncover the origins of activation energy in proteins, and what is its role in facilitating quantum mechanical tunneling? And finally, what is the connection between the thermal activation of enzymatic hydrogen transfer by tunneling and the other classes of enzyme catalyzed reactions? So does tunneling stand in a category all by itself, or is it in fact related to just the general concept of how in the world do enzymes achieve such remarkable rate acceleration? And then finally, this question, which really is fun and we'll talk about later, did the evolution of life depend critically on the observed quantum behaviors in enzyme reactions, or are these a consequence of fundamental principles that underlie all of biological catalysis? Now, PCET, as I mentioned, is widespread, fundamental. Some examples would be photosynthetic water splitting, oxygen reduction coupled to ATP synthesis, et cetera. And many of these large machines catalyze multiple proton and electron transfers. Sometimes these occur in a concerted manner. Sometimes they occur in a stepwise manner. And what I want to introduce today is an enzyme. It's a plant enzyme that functionalizes fatty acids and has emerged as a very beautiful and simplified paradigm for a single concerted proton-electron transfer reaction. So it's PCET in a particular way, which I'll show on the next slide. Now, the beauty of SLO is that it displays enormous room temperature isotope effects. We see in one case an isotope effect of 700, close to what Bell quoted for reactions near absolute zero. They also show very non-classical temperature dependencies. And really SLO, among the proteins that have been studied, has opened up a window into understanding, and this is going to be a key point of today's talk, the role of the protein scaffold in promoting the active site coherence that's required for tunneling. Here's the reaction catalyzed by lipoxygenase. The substrate is a long chain fatty acid, and the carbon-hydrogen cleavage occurs at a center, carbon 11, that lies between two double bonds. That's the donor. The acceptor is an active site iron, a ferric hydroxide. And the proton in this process moves to the hydroxide ion on the cofactor, and the electron moves into the iron. So they're going to different places, but the reaction has been analyzed in detail and is considered as a concerted PCET reaction. So an alternative way of thinking about this is that it's H dot transfer, but the mechanism is truly PCET, and that the proton and electron go to different places. And the intermediate is a delocalized free radical, which then can react with molecular oxygen, leading to the hydroperoxide product. The crystal structure of lipoxygenase has been solved from many, many sources, including the plant enzyme. And this is a picture of the active site. We see a model of a bound substrate with carbon 11 indicated by black. And there's the ferric hydroxide cofactor nearby. This is a model of substrate in the active site. We don't have a crystal structure, but we have other methods of interrogating how it binds, which I also will talk about briefly later. But what I want to point out in this case is the preponderance of hydrophobic, bulky hydrophobic residues in the active site. And the three that I'm highlighting here are two leucines that sandwich the reactive CH bond at carbon 11 on either side of the bound substrate, Lu754 and Lu546. And then one helix turn away closer to the substrate portal is another side chain, Lu553. Now, in this particular study, Matt Meyer made a series of mutants at the remote 553 hydrophobic side chain, as indicated here. So the data are presented in a brief form just to show the energies of activation are fairly small, some of the smallest seen for enzyme reactions. The isotope effects for the series rise from around 80 to 200. But the trend that we picked up and had, fortunately at this point, some background information in terms of how to interpret these data, is that the difference in energy of activation, which is quite small for wild type enzyme, rises in a regular fashion as we increasing perturbation to the interior of the protein by replacing an ILU by a different shape or size hydrophobic side chain. And these data, together with earlier data, have led to the search for a model, a new model of how hydrogen can be transferred in condensed phase near room temperature. And what I'm not going to show any of the math today, but because I thought this would be much easier to explain, given the wide range of research interests of the people present in the audience. And I want to acknowledge Mike Knapp, who used ideas put forth by Kuznetsov and Ostrov to lead to what we call a multidimensional vibronically non-adiabatic model to explain what's going on. And this was picked up by a wonderful theoretician, Sharon Hammer-Schiffer, and modified and refined. But I'm going to give you the big picture here. Basically, a protein is not a static molecule. It's moving all the time. You can almost think of it in some ways as a bowl of jello that's moving around. And so the substrate in there responds to this movement, and some of the complexes will be inactive and some will be where the substrate is properly positioned will become active, and we call that A. And that's kind of like a conformational landscape that leads to these multiple substates. In addition to that, in order to get to the tunneling coordinate, energy has to be put into the system, and that takes us from A to A star. And what this does is it, first of all, puts in it's basically the reorganization energy that comes out of a simpler model for electron transfer, in which, in order for tunneling to occur, we have both a horizontal coordinate where the energy level of the reactant and product have to become transiently degenerate. And then we also have the issue of distance, because tunneling occurs at very short distances, about 2.7 to 2.8 angstroms between the donor and acceptor. But van der Waals distances are out at 3.1 to 3.4 angstroms. So we have this compression and this tuning of the degenerate level up to degenerate levels for reactant and product. Now, when this energy is provided to the system, we reach what is called the tunneling ready state. And this is where we get the transient coherence, in which the wave function is simply delocalized between the reactant and product wells. So the ability to reach the tunneling ready state is temperature dependent for sure, but largely isotope dependent. Now, once we're at the tunneling ready state, the wave function overlap is temperature independent, but it's isotopically dependent. And so because of the difference in mass between deuterium and protium, the wave function overlap is less for deuterium than protium, and therefore the isotope effect reflects this difference in mass in this model. Now, one thing that has needed further elaboration came up in the context of that study I showed you, where we varied the size of a hydrophobic side chain in the interior of the protein. And we moved from this situation where there was very little difference between the slopes of the Arrhenius plots for protium and deuterium transfer into a situation where the plot for protium transfer didn't change that much, but the plot for deuterium transfer became much larger. That is, the energy that had to be put into the system for D-transfer went up. And this is called the donor-acceptor distance sampling mode. And this is quite different, of course, from electron tunneling, where we can't do these kinds of experiments to begin with, and where we're moving a much larger, over much larger distances with a smaller particle as well. Okay, so we have this donor acceptor distance sampling, which explains the trend in the difference in energy of activation. And we have a theory that seems to be pretty all-encompassing, but runs into problems when we work with more adiabatic reactions. And I don't have time to go into that today, but it continues to be an interesting area. Okay, so with that, let's go back to native enzymes and how do we generate an active site that's so compact that it can support detunneling with little additional input of thermal energy activation relative to protium. Now, can the ground state of the ES complex of SLO, is the protein pushing? Is it a rack? And create a tunneling ready state where the donor and acceptor are 2.7 to 2 angstroms away. We would love to get a crystal structure, as I said, of the substrate or substrate analog in the enzyme, but with this enzyme SLO, from SLO, it has not been possible. And we turn to a technique called ENDOR to measure the nuclear electron spin in the ES complex. We can form the ES complex, we just can't get crystals to grow for that form of protein. And from that, infer the donor acceptor distance in the resting ES complex. And this involved a lot of groundwork, synthesizing the substrate with carbon-13, and in particular in the reactive position, substituting the iron with manganese because the iron gave too fast relaxation. And it was a study that I'm very proud of. The lead ENDOR spectroscopist is Brian Hoffman and his co-worker. We also involved Sharon and her co-worker to do the theory, and then two people from my lab, Adam and Marcus, who did the synthesis, enzymology, etc. Anyway, to cut to the chase, these are the results. And basically what we measured was through the nuclear electron spin, the distance between the reactive carbon and the metal, and that's shown in the red line. And we did that both for the wild type and mutant. We had crystal structures of the manganese enzyme where we could get the distance between the metal and the hydrogen acceptor, the water or hydroxide. And then just using the law of cosines, which all of us learned in middle school, we could calculate, of course it was more complicated than that, the distance between the reactive carbon donor, the reactive carbon, the hydrogen donor and hydrogen acceptor. And lo and behold, it's van der Waals. There's nothing rack-like about this protein in a static ground state. And when we make mutants, of course, we could see how the donor acceptor distance moved apart. And this relates to the temperature dependence of the KIE in the perturbed enzymes. So how does the enzyme reduce the donor acceptor distance from 3.1 angstroms to arrive at the tunneling ready state? So let's return to this picture of a conformational landscape where I'm showing a series of coordinates. Normally we just write a substrate going to product along the reaction coordinate. But we have to consider the fact that the protein is not static, it's moving all the time. And this will create a whole a very large number, n possible microstates. And if we consider going from a free substrate to bound, and then let's just consider this is the tunneling ready state, we go down to product and free product. That would mean that there are a whole range of small differences in the activation barrier due to the movement and sampling that's going on. And some subset of conformers is expected to be most optimal for barrier crossings. But note, this conformational landscape, which is considered to be in equilibrium, everything is equilibrated, are generally attributed to relatively low activations of 1 to 2 kcal per mole. That's KT, room temperature. And thermal activations, interestingly, with the exception of the wild type of SLO, are often significantly much greater than 1 to 2 kcals per mole. So that got us thinking about the idea of truly dynamical processes that impact the activation, the thermal activation of enzyme function. And there's been a lot of work, very beautiful work using NMR to actually look, do after peak assignments across the entire protein to look for different substates of protein structures that accompany the transition of substrate to product. And I point out the work by Peter Wright at Scripps in San Diego, who worked on the tunneling enzyme dihydrofolate reductase. But we wanted a more generic method that could use small amounts of protein under the conditions of catalysis that didn't require labeling. And what I want to introduce you to is a tool that we think is extremely informative to understand what the dynamical processes are in a given protein and how those processes can be related to functionally altering mutations, so that we can then make correlations between the impact of a mutation on catalytic efficiency, in this context, hydrogen tunneling, and the impact of the mutation on protein local flexibility, and that's the temperature-dependent HDX-MS technique. Okay, so let's do an overview of what I'm talking about. Here's a protein in solution, moving around all the time, undergoing local protein unfolding at different regions of the protein. And when it does that, it opens the protein up. And if we place the protein into D2O, and note this deuterium now refers to the solvent and not the label on the substrate, with time as the protein opens up, the deuterium can exchange from the solvent into the backbone of the amide. And this technique was pioneered by Walter Englander using tritium initially, but is now a widespread technique in biochemistry and molecular biology. Now, if we wait a different length of time and then quench by going to low temperature and pH and use a pH active protease, we get a set of peptides. We can isolate each peptide and look at the incorporation of deuterium with time. And then the different colors here represent different temperatures. And this allows us to look at differences, spatial resolution of differences in flexibility and how that responds to the gradient of temperature. The expression is very simple. The kinetic expression, the protein must open and then undergo exchange. So K exchange is the intrinsic exchange once the protein is open, but the protein can also close. And this gives rise to the rate constant, the approximate rate constant here. But importantly, we choose conditions where K close is much faster than K exchange. That's called the EX2 regime. And what that means is that the observed rate constant for the rate of incorporation of deuterium into the backbone of the protein is a large K open. It's just the equilibrium constant for this local unfolding, multiplied by the intrinsic rate constant. And therefore, the energy of activation, or the enthalpy that we'll observe, is going to be the sum of the enthalpy for just this equilibrium, opening, closing, delta H naught, plus the enthalpy for the actual chemical exchange. Now, what Adam Offenbacher did was, with SLO, he extended this in a really useful direction. So first, let me say SLO is not for the faint of heart. It's a large protein. When we use the protocols I just showed you, he obtained upwards of 500 peptides. He chose 49 non-overlapping peptides to give 89% sequence coverage of the protein. And he measured the HDX not only as a function of time and temperature, but now a functionally impactful mutation. And he identified the peptides that show an impact of mutation on the temperature dependence of the rate constant for HDX. Well, when we make a single site mutation, this is not going to alter the intrinsic rate constant for exchange, except perhaps in the immediate vicinity of where the mutation is. And so when we look at the differences in energy of activation between a mutant and the wild type, in fact, the intrinsic rate constant temperature gradient falls out, and all we're looking at are differences in local flexibility between a mutant that's impacting function, in this case tunneling, and the wild type. So this really simplifies things. Now, when he was done analyzing the entire protein, what he found were two and only two peptides that showed an impact of mutation on the temperature dependence of hydrogen deuterium exchange. And here they are. This is the full picture of SLO. The iron is right here. The substrate will sit down right in here. And what he picked up were two regions of peptide colored orange and in an orange box. And in fact, we could analyze either one of them. I'm showing you the data for the 317 to 334 region of protein. And what we were able to do was compare then the impact of mutation in this region near the active site. We already knew what those mutations did to the energy of activation for catalysis, that's shown on the right. And in this new series of experiments, we could do the same thing for hydrogen deuterium exchange, and we plotted them, and lo and behold, there's a correlation. The absolute magnitudes of the energies of activation are not the same. These are two different chemical processes. One is active site tunneling, the other is this opening and closing at the protein surface, but the correlation is what's important. If we look at the structure of SLO, we find that this, in fact, defines a network. And the network that we see, the spatial resolution of a network that we get from hydrogen deuterium exchange, is largely hydrophobic. You can see the green balls. The active site, once again, is here with substrate binding between 546 and 754, which is up here. And there's a single hydrogen bond, and that's it in the network, and it goes out to the solvent. Now, we have separated this region, this thermal network, into three regions, which I'll discuss a little bit more as we go forward. We have the active site near the iron. We have what we call a collar region that communicates what's going on in the active site. And then we have the extended network that goes right out to the solvent. I just want to show you that the collar side chains have been mutated, and they reduce the activity of the enzyme. They change the energy of activation. They don't change the temperature dependence of the KIE. And we were able actually to extend the HDX data to room temperature X-ray studies, a technique pioneered by Tom Albert at UCSF and carried forward by Jamie Fraser on the faculty. Tom Albert was at Berkeley. James is on the faculty at UCSF, and Christine Guy is at Berkeley, working in an X-ray facility. So what we did was extend the studies of HDX, and I just want to show you this alternate tool of looking at multiple conformations for protein side chains at room temperature via X-ray crystallography. This is a growing and important area. And if we just restrict our view to the side chains whose distribution of conformers is impacted by the mutations in the collar region of SLO, what we find is something that extends our view that emerged from HDX. And I'll just show you that very quickly. Here's one of the collar regions, ILU 552. it has no effect on 553. That's a control. But if we look at Lu 546, which is near the reactive carbon to substrate, when you make the mutant in the collar region, these alternate conformations go away. Thing dampens down, it clamps down. If we look at the alternate collar region, that is also impacted. So 750 is the other collar region. That also undergoes a change in electron density when we mutate the alternate, the matching pair of hydrophobic side chains in the collar region. And we could go on and on. I just want to highlight the two other side chains that are in the network now that are affected both by 552, where we lose this possibility of a range of alternate conformations. So if we look at the wild type versus the mutant, we're dampening the protein down. And that's also true when we focus on 750 as well as the 552. So I want to show you the full picture that has emerged as a combination of both the hydrogen deuterium exchange and the room temperature X-ray data. Essentially what we've defined is a cone in a very site-selective region of protein that extends from the active site into the protein solvent interface. And we show this in a two-dimensional plot where these hydrophobic side chains, with the exception of this single hydrogen bonding pair, are all interconnected via van der Waals contacts and lead out to the solvent where we see glutamine at position 322. So this is the network we propose that leads from the solvent to the active site. Okay, that takes us to water. And the question is, where does the activation energy come from in biological catalysts? Well, it's got to come from the solvent bath. And the model is that this collisions from the solvent bath lead to restructuring at the protein-water interface, and that this is transmitted in some collective manner to the active site. There's a great deal of work looking at the relaxation that can occur, processes that can occur at a protein-water interface. And this is a beautiful theoretical paper from Bakshi in India, where he's shown that there are two classes of water that have different relaxation rates. The first is the bulk water, which will relax quite rapidly. And then he defines the water that is either bound to the protein or in this zone of biological water that will relax more slowly in the range of nanoseconds. Well, with that information and the definition, the spatial resolution of a network in SLO, it was beholden to us to see if we could, in fact, find evidence for true dynamics, ballistic effects in proteins that can control the tunneling behavior at the active site. And Paulo Zaragoza, a postdoc currently in the lab, took to modifying SLO, making two sites of mutation, one at the end of the thermal network and the other in a control position, 596, putting in cysteine, which he could then modify with the fluorophore Bedan. And he then proceeded to do the measurement of Stokes shifts for this new probe, either at the end of the network or at the control position. So what is a Stokes shift? It's not really that complicated. Essentially, you have a fluorophore. You photoexcite it, you go to an excited state dipole, and then the environment, and you have a certain emission. And then the environment starts to rearrange around the excited state dipole to stabilize it. The excited state dipole is very dramatic in Bedan. And what we have is an environmental reorganization that stabilizes the dipole and then leads to the emission of the light at a longer and longer wavelength. And so you just follow they're called Tress curves, the change in the lambda max emission as a function of time, and you can do that as a function of temperature. And we found two transients. One was about half a nanosecond, which is more similar to the bulk water, and then we had a dominant one at around 5 to 7 nanoseconds. And we plot the temperature dependence of the larger amplitude Stokes shift as a function of temperature for the control site and for the network that we've identified. And lo and behold, these are the data. Now, essentially what we're seeing is the same energy of activation for KCAT in the buried active site as the environmental reorganization for the BEDAN that's sticking out into the solvent when we attach the BEDAN to the end of the network. But when in fact we put the BEDAN on the control site at the other side of the protein, there's no correspondence. Well, this led to the idea of a thermal quake, if you will, or protein quake. These ideas have been put into the literature by other very prominent scientists, but the ability to actually relate the quake at the solvent interface with a tunneling phenomenon in the active site is new. And what we propose then is that the collisions of water with the protein surface give rise to a concerted cooperative movement in this region that equally affects the reorganizational energy around the extended chromophore and provides the energy that's needed to provide the tunneling ready state buried in the active site. At some earlier data, and this is a totally different hydrogen transfer system. It's a hydride transfer. It's called a dehydrogenase. It's a tetramer and not a monomer. And many years ago, we had shown it's a thermophilic enzyme. Its behavior breaks at 30 degrees, and the physiologically relevant regime is above 30 degrees, and below 30 degrees, the protein rigidifies. But what we saw at that time, and we didn't even understand it, was this temperature independence of the KIE, that's these data in the squares. It's temperature independent in the physiological range. The slopes are close to identical, and then it starts to rise when the enzyme is impaired, similar to what I've just explained to you for SLO. We also did HDX in the much earlier, and this was in fact our first HDX study in collaboration with Natalie On at University of Colorado Boulder, and we found this is the cofactor binding domain, this is the substrate binding domain, and we found a trend, a break in temperature with HDX that correlated within the substrate binding domain with the break in catalysis. Now, this enzyme is remarkably amenable to looking at fluorescence quenching or FRET, or fluorescence lifetimes, fluorescence quenching in FRET or fluorescence lifetimes because of the presence of only 3 tryptophans, 2 within the substrate binding domain and 1 way out in the cofactor binding domain away from the active site. And we were able to engineer protein with single tryptophans at each of these positions, and then look at the impact of the temperature on either the fluorescence lifetime, the Stokes shifts, or FRET. So the W167 is the control, and the 2 catalytically relevant tryptophans, which are within the active site, is either W49 or W87. And so when we look back and compile on the data, we had something that struck us as really meaningful. We have, in fact, the energy of activation for catalysis for the variant in which we have a single tryptophan. And then if we look at the fluorescence lifetimes, same energy of activation. We have the Stokes shift, similar, not exactly the same. And that's for the enzyme containing the single tryptophan W87N. If we do a similar experiment for 49, W49N, in that case, we do a FRET between the tryptophan 49 and the bound cofactor, the NAD. We get the same energy of activation, whether we look at the temperature and time dependence of FRET and catalysis, or fluorescence lifetime and catalysis, et cetera. And when we go to the control, which is out on the other side of the protein, there's no correlation again. So this, together with the HDX, has allowed us, the HDX shows the separate networks within the substrate binding domain. And then the fluorescence analyses indicate similar thermal quakes, if you will, this ballistic effect coming in from several directions, initiating at the solvent interface of the protein and culminating in the active site. Now, one thing I want to just point out before we move on is that the lifetimes are different, clearly, for catalysis, which is millisecond. For a Frasch experiment, the T-jump was microsecond, and then the Stokes shifts, which are nanosecond. So how can we think about this? We have the same energies of activation, but different times. And so what this means is that the processes that we're looking at, of course, have two components. The first, of course, is the intrinsic rate, which has this thermal activation associated with it, and which we're attributing in these cases to these localized protein quakes. But the rate, the times for the observation depend on the probability that the quake will create the environment that is amenable to what we're measuring. So for environmental reorganization out on the solvent surface, we may not need that precise an orientation of the solvent water in relation to the Badan, and the impact on K observed of this fraction of protein that's in the right conformation can be pretty small, and we can get a very fast rate constant. For Frasch, we have to get the donor and acceptor in the right orientation, much more demanding, and then the impact of the fraction of enzyme that reaches the right conformer is much greater. And voila, catalysis, for catalysis to occur, so much has to be optimized in that transient time that, in the case of tunneling, for example, the degenerate states are achieved the right distance between the donor acceptor for appropriate wave function overlap. So you can see we can easily rationalize identical energies of activation for a range of processes that we can relate to K-cat, but with different time scales. Okay, so at this juncture, here's a model. And the model is that these proteins are not static, and that the proteins clearly undergo this equilibrated conformational sampling, which can, in fact, in the presence of substrate, prejudice the protein toward an orientation of the bound substrate into a position which favors the acceptance of this thermal activation through these encoded, evolutionarily encoded thermal networks that are part of the protein structure / dynamics. And then, of course, we have these kinetic activation processes in which we can introduce enough heat to give rise to reaction, even with energies of activation that are significantly larger than room temperature. So this is a two-state process. Now, to finish up, how am I doing time-wise? What I want to do is just move from a tunneling system to a more generic analysis. I've shown you either lipoxygenase, which catalyzes PCET, or dehydrogenase that catalyzes hydride transfer. I just want to show you our recent results of the generality of these thermal networks. And so this is a study that we began 3 years ago, where we decided to look at a common scaffold in biology. It's called the Tim barrel scaffold. That's been enormously well studied, and it catalyzes 5 out of the 6 total reaction classes in enzymology. But it's been shown that the active sites are all within the center of the barrel. It's a series of helices and sheets. The position of the active site changes for these 5 classes of reaction. And we wanted to know, do the thermal networks undergo coevolution with changes in the chemical nature and geometry of these states? And I'll just show you briefly our flagship study to give you an idea of how this was carried out. This is the enzyme adenosine deaminase. It's a single subunit enzyme. We're looking down into the barrel on the left and on the back of the barrel on the right. And this result was just published at the end of last year in JACS. So basically, adenosine deaminase, even though it catalyzes a proton transfer, is a class of reactions that's totally different from what we've been looking at with tunneling. And in this case, we're going to take the adenine ring of adenosine, and in the presence of an active site metal, which has water on it, we're going to use a base and an acid to add water into the carbon as indicated in the adenine ring to form an intermediate, which then breaks down and releases ammonia. So this enzyme is very well studied. It has an active site metal with ligands, 3 histidines, and an aspartate. It has a base, his-238, an acid, glue-217. And here's a transition state analog, whatever that means, a tightly bound inhibitor. And what we identified was a hydrophobic wall, basically, behind the adenine ring of the inhibitor. And we focused on Phe61, which is one of a series of 4 hydrophobic residues in this region. And this is seen to be within van der Waals distance again of the bound analog. And we then proceeded using our tool of mutagenesis to make mutants at that single site, altering the packing within the interior of the protein. And we measured the change in energy of activation as we change the size of the residue and get increasing impairment. And you can see that the energy of activation in the wild type is about 12 kcals per mole. And as we make mutants, it goes up as high as 20 kcals per mole. And so we have a beautiful trend, and we can now, of course, look at what happens to the flexibility of the protein as a function of time, temperature, and mutation. And we chose two. We chose the Phe61 Lu and the Phe61 Ala. And we chose two for the following reason. These mutants change the rate as well as the energy of activation. And so we thought by using the Phe61 Lu as a control, we could look with Phe61 Lu has the same energy of activation as wild type, but the reduction in rate for the Lu substitution is the same as the reduction in rate for the Ala substitution. So if we could compare wild type to Lu and a wild type to Ala, and then Lu to Ala, we could subtract out any changes in protein dynamics and flexibility that are focused on this probability of catalysis, the kcat, and focus on the almost 12 to 17, 5 kcal difference between, 12 to 17, 5 kcal difference between the wild type and the Ala. All right, so let's look at the data. These are bar graphs just showing the peptides that have been analyzed. We have close to 90% coverage of the full protein once again, and the comparison of the energy of activation for hydrogen deuterium exchange in the wild type, the mutant of interest with the large difference in energy of activation, the alanine substitution, and the control, which is shown in gray. That's the ILU. And after we correct for peptides that have the same energy of activation for the ILU and the Ala mutant, meaning that these are not the ones that affect the energy of activation, we get the following result. And that's shown with the arrow here. Now, this shows a network going once again from the solvent into the active site. And the Phe that we mutated is out here. These are heat maps. The darker the pink, the larger the impact. We have the active site metal, the acid, the base, the ligands to the active site metal, all within these thermal networks. And we also saw a fainter thermal network involved in interaction with the remote ribose ring of the adenosine itself in the bottom. Okay, so we have now, in utilizing the soybean lipoxygenase data, for which we have extensive information, and the thermophilic alcohol dehydrogenase, these are the two hydrogen transferring tunneling enzymes that I spent most of the time talking about. We now have, in fact, thermal networks for different classes of enzymes. I already showed you adenosine deaminase. Yeastinolase is another member of the Timbaro family. It's a dimer rather than a monomer, and it also has a large loop closure. So yeastinolase is quite different structurally and mechanistically from adenosine deaminase, yet we see a thermal network, and we see that the nature of the thermal network, which is primarily indicated as A and B. Here, where the active site is this cherry red, and that's where the loop closes over the active site. The yeast enolase thermal network is unique from adenosine deaminase, and the lipoxygenase thermal network is unique from the thermophilic alcohol dehydrogenase. We believe that in each enzyme, whether it belongs to different scaffold or within a family of common scaffolds, has evolved a unique thermal network for the ballistic activation of active site chemistry. Here, this is the adenosine deaminase shown again. You see that all of the thermal network is in the front face of the barrel. The back face is not really affected by mutation, and you can then see that this is basically a more rigid scaffold, which does support then the very dynamic and squishy interior of the protein. And here's a recently published work of a methyltransferase, certainly not tunneling reaction, in which we have a network that leads both along the horizontal and vertical directions in terms of the distance sampling and the adjustment of the position of the donor and acceptor, as well as the electrostatics. So we see a unique thermal network there as well. Okay. So I would say this idea of thermal activation via genetically encoded thermal networks acts in concert, of course, with this lower energy conformational landscape, which is going to be adaptable to the presence of substrate and even allosteric ligands as a generic way of thinking about how these reactions take place. Okay. So this leads us to, we've answered a lot of the questions that we started out interrogating. We can see how the protein scaffold and its dynamical properties lead to coherence and can give rise to the properties that we've been able to elucidate for the CH activating enzymes that work by nuclear tunneling. But there's really, in a way, a truly fundamental question. I call it the chicken and egg question. And this gets back to the finding of these closely packed tunneling ready states that we can deduce from the tunneling properties of modern CH activating enzymes, right? So is this a consequence of a direct role for nuclear tunneling in driving the evolution of biological fitness? Or is the prevalence of tunneling in enzyme reactions a consequence of the fundamental properties that underlie the evolution of optimal biological catalysis? And quantum tunneling just comes along for the ride because the properties of optimal catalysis happen to coincide with the dictates of quantum tunneling. Now, in the latter case, tunneling has been, I think, an important driving force. Tunneling has been an important driving force in allowing us to understand the origins of the enormous rate accelerations, but it may not have been a major driving force in driving protein evolution. So to have some fun at the end, I wanted to do some speculation, because if tunneling really did play a role in the evolution of enzyme function, and if, removal of protium and deuterium from highly reduced compounds that could serve as an energy source played a role, we have to think about the ratio of deuterium to protium in the universe. And it turns out deuterium is a very rare element because under the conditions of the Big Bang, I'm not a physicist or an astrophysicist. I just have been doing some what I call armchair reading in this area. But certainly under the conditions of the Big Bang, deuterium disappears quite rapidly because it undergoes conversion to helium. Protium is stable. And astrophysics is very involved in trying to understand the ratio of deuterium to protium, especially in understanding the origins of water and life. And so here are some numbers. On Earth, we have 26 deuteriums per 10 to the 6 hydrogen for an abundance of 0.02 to 0.03%. Comets can be enriched tenfold, and Venus is enriched 100-fold. And these are all very low ratios, and they're actually higher than expected. And there's been speculation there may be enrichment of deuterium in dark matter. Now, with such low levels of D in the universe, how could the evolution of enzyme activity have been driven by the need to create compact active site geometries that are able to support deuterium tunneling with activation energies similar to protium tunneling? I don't know. But here is where the fun and speculation aspect comes in. And one possibility is that the D to H ratio in the early universe was actually much higher than currently estimated. It's probably completely off the wall, but worth putting down. And the other possibility is that under the conditions of the evolution of life, there may have been extraordinarily low levels of reduced compounds that could act as a food source. And this would have led to the importance of utilizing both compounds with protium, but also the trace compounds with deuterium, with the species that could utilize the foodstuffs with deuterium surpassing the competing species. And I'll leave you with that fun topic and just this has been a very long, many decades effort. And I just want to highlight Adam and Pello, who did a lot of the recent work on SLO. And of course, our wonderful collaborators, Sharon Hammer-Schiffer, Tom Alber, who sadly is now deceased, Sharon, Brian Hoffman at Northwestern, Jamie at UCSF, and the people at UCB. Amnon Cohen started the thermophilic ADH project, and that was carried into the more modern studies of fluorescence, lifetime, Stokes shifts, and FRET by Corey and Jianyu in collaboration with Tom Spiro and Brian Dyer. I also want to acknowledge Natalie Allen for her help in enormous contributions to our laboratory being able to pursue the hydrogen deuterium mass spec study. She's one of the real pioneers in this area. And then the Timbaro enzymes, all the recent work by Emily and Schwaiwa, and the methyltransferase work with Jianyu and also Natalie On. And I thank you. All right.